Welcome back, and let's talk today about the fertilization. But before we do so, I would like to talk about the female reproductive system. You've been given these slides maybe earlier this semester. However, today I will take you through some of these important parts. This is the vagina, where the sperm by the end of intersexual course is deposited or are deposited. Then they travel from the vagina through the uterus. What you need to know about the uterus, that it has a cervix, it is closed, and then a cavity, the body of the uterus here, fundus, and most importantly today we will absolutely talk about the uterine tubes, also named oviduct because they carry the ovum, or fallopian tube. They are 4 inches or 10 centimeters in length. They are quite long, and they lie in what we call the broad free edge of this structure, the broad ligament. Sure, I'm not gonna ask you in the exam about these details because we will talk about them next in the course of UGS system, urogenital system, by the end of the 3rd year. So what I really need you to know, that they are the root for the sperms, as you can see. I'll draw some pictures here. Now sperms are deposited here, then they travel through the uterus until they reach the uterine tube, and they meet the ovum in the ampullary part somewhere here. So that's why we need to know everything about the uterine tubes, as they play an important role in fertilization, especially if we know that the sperm meets the oocyte in the ampullary part of the uterine tube. So their function is in transportation of the sperm first of all, and then after fertilization they help in carrying back the fertilized egg to the uterus. The uterine tube has 4 parts. I'm sure the first one here is the they are here. First one is the infundibulum. As you can see here, it's the part that communicates with the abdominal cavity. It's the closest part to the ovaries. It has ostium here, and this ostium has these finger-like projections. We name them fimbria. Before ovulation, these fimbria, finger-like projections, they sweep over the oocyte, the ovaries, to catch the ovulated egg. The second part is the ampulla, and the ampulla is very important simply because I'm not sure this is the right indication of the ampulla. The ampullary part somewhere here. I'm sure it should be somewhere here. There it is, the ampulla. It is close to the infundibulum, not far away from it. Anyway, the ampullary part, then the isthmus here, and finally the interstitial part, which is the last part before where it joins the uterus. So I will be interested in this part, the ampullary part, where the fertilization takes place. So I was talking about the female reproductive organs. and I focused on the uterine tubes. You need to know their parts, and you need to focus on the ampullary part of it, and of course to know that the infundibulum is in a close relation to the ovaries, and it catches the ovulated egg through its fimbria. Finger-like projection, they sweep over the ovary to catch the ovulated egg. Now, if you take a cross-section inside the ovary in a uterine tube, you will of course see that the uterine tube has simple columnar ciliated epithelium. This is the cilia, and I'm sure that in histology they talked about this. This cilia helps in the movement of the oocyte. So let me just remind you that the oviduct plays two functions. The first one, it facilitates the movement of the sperm. The sperm passes through the uterine tube until it reaches the oocyte, where fertilization takes place in the ampulla. Then the fertilized egg, now the fertilized egg would go back towards the uterus, and what would help the fertilized egg to go back to the uterus is the peristaltic movement of the walls of the uterine tube, peristaltic movement of the uterine tube, because the uterine tube has in its walls smooth muscle, and they rhythmically contract, and they move the fertilized egg towards the uterus. Also, what helps the movement of the fertilized egg, of course, are the cilia of the simple columnar ciliated epithelium of the uterine tube. So as you can see, you need just to memorize the things in the boxes. They are easy. And now let's talk about the last product of the last lecture. In the last lecture we talked about oogenesis, and we have reached the puberty, and we talked about it in details, and we said that the primordial follicle, under the influence of FSH, it becomes primary follicle, and this primary follicle becomes secondary follicle, and then under the influence of LH, luteinizing hormone, it would become a tertiary follicle, also called Graafian follicle, that we are looking at now. So the changes were all about the follicle itself, the wall and the thickness, antrum, and etc. And we have reached this Graafian follicle, which contains the oocyte, and the oocyte just finished its meiosis 1. So this structure has been named cumulus ovoris. Cumulus ovoris. Now cumulus ovoris has inside it granulosa cells, zona pellucida, a secondary oocyte arrested in metaphase stage of meiosis 2. So you have to understand that we have started with primordial follicle. What was inside it? Primary oocyte that have initiated meiosis 1, and it was arrested in diplotin stage. Then it took us long time during menstrual cycle from primordial to become primary, secondary, and tertiary. In the tertiary follicle, in this Graafian follicle, the oocyte under the influence of LH has completed its meiosis 1, and as a result we now have two cells, big one, the secondary oocyte, and in its circumference here, in its poles, swimming another small polar body named first polar body, and that's why we have written here first polar body. And the name polar, it means that it is within, not far away from the oocyte. I have other drawings, we will talk about it in just moments. So as you can see, we didn't reach the final stage of the oogenesis. What we have here is a secondary oocyte that have initiated meiosis 2, and it is again arrested in meiosis 2 in metaphase. So we didn't finish meiosis. So today we will finish this stage. Now we need to talk about pre-ovulatory or Graafian follicle, how it will be ovulated. So today we will talk about ovulation. LH increases. We have reached day 14 of the menstrual cycle, and LH increases. The surface of ovary bulges locally, so under the influence of LH, the wall of the ovaries bulges locally. So we are talking about this here. The wall here in this area of the ovary would bulge because under it there is Graafian follicle that needs to rupture. Digestion of collagen around the follicle, the part of the this wall that surrounds the follicle would be destroyed by the by enzymes, and then local muscular contraction on ovarian wall would take place. So we are talking about this area here. Let's do some drawing here. This area, we have a follicle here, we have a Graafian follicle, big Graafian follicle, okay, and it has to go outside to be ruptured outside, and all this is happening under the influence of LH. So LH that has been secreted from the anterior lobe of the pituitary gland, it would reach the ovaries, and it would influence its wall, and the wall should be broken in this area, and as a result all this will help in extruding the cumulus ovoris to break free, and this is what we call ovulation. This is what we call ovulation, and it float out of the ovary. So this takes place at day 14 of menstrual cycle, and what is interesting that the ovaries, that ovulation, it does not mean breaking through free for the whole Graafian follicle. Only the cumulus ovoris would be ovulated. So what would go out of the ovary is just this, and it would leave behind it all this. Theca externa, theca interna, they will stay in the ovary. So what would be ovulated really is the cumulus ovoris, and not only cumulus ovoris, but you need to understand cumulus ovoris has inside it granulosa cells around it, zona pellucida, secondary oocyte, and first polar body, but they are making a small part of the Graafian follicle, and they would be ovulated out. So ovulation. Now day 14 of 28 menstrual cycle. Menstrual cycles varies in their length. They can be short as 21, and we regard this as a normal menstrual cycle, and they can be as long as 35 days, and we also regard this as menstrual cycle. However, having said that, the average is 28 days. So usually we would talk about 28 days. Mid-cycle, it means day 14, but it is mid-cycle for other normal variations. If it is 34, 35, then day 17. If it is less, then we just take the half of the cycle. For you, you need just to stick to 28 days. Mid-cycle, it is day 14. So on day 14 of the menstrual cycle, ovulation would take place. The cumulus ovorus would leave the Graafian follicle free, and it would be catched by the fimbria of the infundibulum. So as the cumulus ovorus is getting emerging from the ovaries, some of the granulosa cells, look back here carefully, these cells, granulosa cells that have been set free from the Graafian follicle, they follow the oocyte, and they rearrange themselves around the ovulated egg, and they would look like rays of the sun or corona. Unfortunately, I'm sure that all of you are suffering sitting at home from coronavirus. Here we have another structure in our bodies, females' bodies, that looks like that it has a crown. The granulosa cells, they rearrange themselves around the ovulated egg, and they look like a crown, and that's why we call them corona radiata. Radiata, it means like rays. They are everywhere. So now, from now on, if I to ask you what has been ovulated, you should answer this. It is cumulus ovorus, but from now on it's not cumulus ovorus, it is the secondary oocyte arrested in metaphase stage of meiosis II, plus its first polar body. Look, where is the first polar body? And that's why we call it polar body. It is like the earth and the moon. They are swimming, the moon is swimming around the earth, and this is exactly a good similarity here. The first polar body is within the nearby the oocyte. So granulosa cells, now we call them corona radiata and zona pellucida. So what has been ovulated is the secondary oocyte plus first polar body, and they both surrounded by zona pellucida, and zona pellucida is surrounded by corona radiata. Why we are talking about this? This is very important. The sperm has to pass through the corona radiata, then it has to pass through the zona pellucida to reach and to fertilize the ovulated egg. Now, as I told you, only from the Graafian follicle, only the cumulus ovorus has been ovulated. If you go back, only this part has been ovulated, and it's interesting to talk about what has been left. So if you go back here, what has been left here is granulosa cells, theca interna, theca externa. All this has been left inside the ovary, and the question is why? Now, after ovulation, granulosa cells remaining, the granulosa cells remaining here in the wall of the ruptured follicle, together with cells from the theca interna, are vascularized. No wonder vascularized, it means they get blood supply. The ruptured follicle, it means bleeding. So they are vascularized by surrounding vessels nearby, and under the influence of LH, these cells develop a yellowish pigment and change into lutein cells. And the name luteinizing hormone is coming from this structure, lutein cells, which form here the corpus luteum. Corpus means body. Corpus luteum, and secrete estrogens and progesterone, mainly estrogen here, and then we will talk about progesterone. It would be to change from now and then. We will talk about this. Estrogen and progesterone together with some estrogen cause the uterine mucosa to enter the secretory stage in preparation for implantation. So I understand that this could be hard for you at this stage, but you need to realize that only the cumulus ovoris has been ovulated. What has been left now under LH would turn into corpus luteum. This corpus luteum would secrete progesterone. Now, progesterone would affect the uterus, and the uterus would get bigger. The endometrium becomes bigger and bigger, the functional layer, we will talk about it later, to receive the embryo. So this is an important thing to know. Now let's go back to the what is the destiny of the ovulated secondary oocyte arrested. You just need to know all this. What is the destiny? What would happen? Until now, the secondary oocyte didn't finish its meiosis II. Now, let me remind you of what we have by now. By now we have the ovulated egg surrounded by zona pellucida plus corona radiata. We already talked about this. Now, is this oocyte ready to be fertilized? Chromosomal wise, no, it's not. Why? Simply because it didn't finish meiosis II. What does this mean? It means that inside this secondary oocyte there is 46 chromosomes, or 23 chromosomes, double structured chromosomes. So if the sperm gets inside, we would have a wrong number. We will not have 46, we would have more than that. So I would say that the oocyte is not ready for fertilization. Let's leave this and go back to the sperm. How about the sperm? Now the sperm that has been ejaculated should be ready. It has passed from spermatogonium and the basal compartment of the seminiferous tubule, as we mentioned. Then it becomes primary spermatocyte, 2N double structured chromosomes. Then it finished its meiosis I and gave us secondary spermatocyte, 1N double structured, then gave us spermatid, which is chromosomally 1N 23 single structured chromosomes, and then it went through spermiogenesis, as we mentioned earlier, and it became, as you can see, it has head, neck and tail. It has everything. So if we compare the oocyte at this time and the sperm, it seems that the sperm is ready chromosomally, and the shape are ready. However, having said that, you need to understand that on the top of the sperm there is glycoprotein coat and seminal plasma proteins. These will prevent, as you can see here, they will prevent penetration through corona radiata. So again, chromosomally and the shape, it seems that the sperm is ready for fertilization, but it needs to get rid of this coat. How? I'll tell you just in a moment. So it seems to me that both the oocyte and the sperm, they need some help. Let's have a look how we're gonna help them to meet each other and to fertilize. And for this, let's go back to day before ovulation, day 13, day 12, 13 of menstrual cycle. Fimbria, you already know this finger-like projection of the uterine tube, sweep over. They sweep over. They go there and there, sweep over the surface of the ovary, and the tube itself begins to contract rhythmically. So in preparation, the tube also begins to contract rhythmically, and the oocyte transport now. These sweeping movements of the fimbria and the motion of cilia on the epithelial lining carry the oocyte on day 14. When the oocyte, the cumulus ovorus is ovulated, it should be caught by the fimbria, and the fimbria would take it to the infundibulum, and the infundibulum from there the cilia and the rhythmic contraction of the smooth muscle in the wall of the uterine tube would carry on the secondary oocyte to reach this area, the ampulla. Now, in humans, the fertilized oocyte reaches the uterine lumen in approximately 3 to 4 days. We will just after fertilization, but we need to talk about this in a moment. Now, fertilization. A process by which a haploid male gamete fuse with female gamete to give single diploid nucleus. This is the definition of fertilization. So, do we have haploid male gamete? Yes, we do. The sperm is a haploid male gamete ready, but it has on its head some plasma proteins. We need to get rid of them. Apart from that, it's ready. Do we have a female gamete haploid? Unfortunately, we need to work on the oocyte. It's not haploid yet. It needs to finish its second meiosis. Where fertilization takes place occurs in the ampulla, again, ampulla region of the uterine tube. Why there? Because this is the widest part of the tube. Spermatozoa may remain viable in the female reproductive tract for several days, and we may talk about this later. Only 1% of sperm deposited in the vagina enter the cervix, where they may survive for many hours. The trip from the cervix to the oviduct requires a minimum of 2 to 7 hours, and all this to fertilize the egg. So spermatozoa are not able to fertilize the oocyte immediately upon arrival in the female genital tract, but must undergo capacitation and a chromosome reaction. So I told you that the sperm on its head there is plasma membrane, and it has to be removed, and this is what we call capacitation. So the sperm needs capacitation, then the sperm needs to go through a chromosome reaction when it contacts the oocyte itself, penetrate the zona pellucida to fertilize the egg. So capacitation is a period of conditioning in the female reproductive tract. In human lasts approximately 7 hours, and it includes removal of the glycoprotein coat and seminal plasma proteins from the plasma membrane that overlies the acrosomal region of the spermatozoa or spermatozoon. Now let's go back to this drawing here. I told you that the sperm has a coat here of plasma membrane. However, as the sperm passing from this area, from here until it reaches the ampulla, within this time takes a long time, from 2 to 7 hours. The female genital system, the uterus and the uterine tube, all these parts of the female reproductive system, they help the sperm to get rid of this cap. So why? We already mentioned in the last lecture that here we have acrosomal cap. What is the acrosomal cap? It is enzymes. They will digest the zona pellucida. So we need to remove this layer, and what would remove this? The female reproductive system, the uterus, the uterine tube, they will remove it, and this process we call it capacitation. So what is capacitation? It is removal of this cap from the head of the sperm. Where it did occur? It occurs in the reproductive system of female reproductive system. So only capacitated sperm can pass through the corona cells and undergo the acrosome reaction. So as you can see here, if this sperm immediately reaches the oocyte, it would be trapped by corona radiata. It is like a wall for uncapacitated sperm. So any sperm that is not capacitated, what does this mean? Any sperm with this layer on it would be trapped by corona radiata. If this cap has been removed in a process called capacitation, the sperm easily can pass through corona radiata. So if I to ask you how the sperm passes corona radiata, the answer should be by capacitation only. Right, now the sperm has been capacitated, the proteins from the head have been removed, and the sperm is ready to face the second wall, which is the zona pellucida. Here, as you can see, this layer has been removed, has been removed, and now the sperm has been exposed, and here we have the acrosome related reacted sperm, or the acrosome, which contains enzyme to destroy the zona pellucida. So this sperm won't pass corona radiata. Only this would pass corona radiata, and only this can act on the zona pellucida. Now, once the sperm have reached zona pellucida, here we initiate the acrosome reaction. Occurs after binding to the zona pellucida, is induced by zona proteins, includes the release of enzymes needed to penetrate the zona pellucida, including acrosine and trypsin-like substances. Now, more on this here. Phases of fertilization. The first one is penetration of corona radiata. The second one is penetration of zona pellucida, and the last one, fusion of the oocyte and the sperm. So we have walls to go through. The first wall is corona radiata. I already told you who gonna pass through it. Who gonna pass through it is the capacitated sperm. So capacitated sperm freely pass through the corona radiata. Now let's talk about the solid rock wall, which is zona pellucida. Now penetration of zona pellucida, the zona is a glycoprotein shell. We already talked who made it. It's a contribution between granulosa cells and the oocyte itself during the process of primordial follicle becoming primary, secondary. We had there the zona pellucida taking place and formed at that time. So it is a glycoprotein shell surrounding the egg that facilitates and maintains sperm binding. Now this is very interesting. I just mentioned minutes ago that zona pellucida is a wall, and by default wall it means that prevents sperms from getting inside the oocyte. However, you need to understand that zona pellucida has more functions than this. Zona pellucida in fact facilitates. It has areas designed for the sperm, but they will not allow more than one sperm to get inside the sperm. So they are inside the oocyte. So they are facilitators. It is zona pellucida. It is a facilitator, and at the same time it prevents. It is a wall in front of polyspermia. It allows only, it facilitates the entrance of only one sperm. Now, the sperm, as it reaches zona pellucida, it binds to ZP3 receptor. This is a zona protein. Then release of acrosomal enzymes. We did mention that the sperm itself has here acrosome, and during spermiogenesis we did mention that the sperm would go through spermiogenesis period where it acquires acrosome and it acquires flagella, mitochondria, whatever, and this acrosome has in it the enzymes acrosin, which allows for the sperm to penetrate zona pellucida. Now, permeability of zona pellucida changes when the head of the sperm comes in contact with the oocyte surface. This contact results in the release of lysosomal enzymes. Now let's go back. As you can see here, this is a sperm trying to hit the zona pellucida, and here it is in its way. Now, the moment the head of the sperm reaches the oocyte, immediately the oocyte itself releases lysosomal enzymes. But before that, the sperm, as it tries to penetrate zona pellucida, it was releasing from the acrosome enzymes. So that's why I've wrote here from the sperm acrosomal enzymes. Now, once the head of the sperm managed to get inside through the zona pellucida, it reaches the oocyte itself. Now, the oocyte responds by releasing lysosomal enzymes from cortical granules lining the plasma membrane. So the plasma membrane of the oocyte contains these granules. Okay, as they get in contact with the head of the sperm, these enzymes are released and they alter properties of zona pellucida. So they are released from the oocyte and they go to the zona pellucida, and they tell zona pellucida to do zona reaction to prevent sperm penetration and inactivate species-specific receptors sites for spermatozoa on the zona surface. It once the needed sperm has reached the plasma membrane of the oocyte, now the oocyte would release from its cortical granules lysosomes that would go back to zona pellucida and tell her to shut down the doors in front of any coming sperm. That means zona pellucida facilitated the entrance of one sperm and prevented polyspermia. So the first phase is easy, capacitation. Capacitated sperms go through the corona radiata easily. The second wall is acrosome reaction from the enzymes of the sperm. They destroy the zona pellucida, and once they have reached the oocyte plasma membrane, the plasma membrane, the oocyte secretes enzymes, and they shut down zona pellucida and they prevent polyspermia. Now the last phase is fusion of the oocyte and the sperm cell membranes. Actual fusion between the oocyte membrane and the membrane that covers the sperm head is accomplished at the acrosomal reaction when the plasma membrane covering the head cap disappears during the acrosome reaction, and as a result the sperm would get inside. It loses its tail. I think in this here it would lose its tail, and other sperms can't get inside, so only the nucleus of the sperm would get inside the oocyte. Of course, other supportive mechanisms would also be there, but the tail would be lost because we don't need it. The sperm does not need it anymore. It was needed to move from the deposited area until it reaches the ampullary part where the fertilization takes place. Now, what we have inside here, we have male pronucleus and female pronucleus. Now the question is, what happened to the secondary oocyte? What happened to the first polar body? The answer is could be put in this way. The responses, egg responses to the entrance of the spermatozoon in three ways: cortical and zona reaction. We already talked about them in details. I'd already talked about polyspermia, so this is a repetition to what we talked about, and this is what is important for us. Resumption of the second meiotic division. Resumption of the second meiotic division. So as you have, as I told you, until now, this oocyte named secondary oocyte, it was arrested in metaphase of meiosis II. So it is the second time that the oocyte is arrested. The first time was in the diplotene stage of meiosis I, from puberty and from birth until puberty for each individual oocyte. This period could be 40 years, could be 50, could be 35, plus minus. And the second arrest is during this in the secondary oocyte. So the entrance of the sperm gives the order to the secondary oocyte to finish its meiosis II, not only to the secondary oocyte but also to the first polar body. So we had here we have two cells, the secondary oocyte and a smaller first polar body. Now the entrance of the sperm would end second meiosis, and that is true for the first polar body, and that is true for the secondary oocyte. And as a result, instead of the secondary oocyte, we would have fertilized egg. We don't have tertiary oocyte. You need to understand this is very important. So we have primary oocyte, we have secondary oocyte, but we don't have tertiary oocyte. We do have fertilized egg. That's it. So what would happen to the chromosomes? Second meiosis, it means splitting of sister chromatids again. What would happen that 23 chromosomes will stay in the fertilized egg. We call them female pronucleus, and the other 23 chromosomes, they would go and form second polar body. So the entrance of the sperm inside the secondary oocyte would result in fertilized egg, zygote, or unicellular embryo, all the names are true, and second polar body. What has happened to the first polar body? The first polar body also finished meiosis II, and as a result it has two secondary polar bodies. The destiny of these secondary polar bodies, usually they would be digested by the embryo, or they would vanish from there, and we would talk about the zygote. So only now the oogenesis of the oocyte has come to its end. So that's why when I talked about the meiosis, I did mention that meiosis is totally different from mitosis. Meiosis is an art. Begins as oogenesis in females, begins from early stages in development, yet in the uterus, and after birth we have primordial follicle inside it, primary oocyte that have initiated meiosis I but is arrested in diplotene stage of meiosis, of prophase I meiosis I, and this would carry on until puberty. Now why? Because physically females are not ready to become pregnant at this age. When they reach puberty, their bodies seems to be biologically ready for to carry the baby, and after puberty, menstrual cycle is driven by two hormones, LH and FSH. FSH would take care of follicles inside the ovary, and they would be transformed from primordial, primary, secondary, tertiary. Now the tertiary follicle would have in it inside it the secondary oocyte. So the journey is at least 14 years for any possible oocyte to become secondary oocyte. At least it needs 14 years. So if we take an oocyte from an ovary of a girl that she is under puberty, we will not be able to fertilize this oocyte, because this oocyte simply has more chromosomes that we do need. It's not haploid until now, and this is a kind of protection for girls. Then each month in this menstrual cycle we have at mid-menstrual cycle, day 14 for a normal regular menstrual cycle, we would have ovulation. What would be ovulated is nothing but cumulus ovoris, and it would be rearranged corona radiata, zona pellucida, secondary oocyte that has initiated meiosis 2 and is arrested in metaphase 2. So yet it's not finished the meiosis, and it would stay arrested in there, and of course it has its first polar body. Now if there is intersexual course and if there is a sperm, the sperm would meet the oocyte. As the sperm enters the secondary oocyte, we would have here fertilized egg plus second polar body. Those are products of the secondary oocyte, and from the nearby first polar body we would have also two secondary polar bodies, but so we can have three of them. Sometimes we have only one. What does this mean? It means sometimes that the secondary oocyte does not divide, so we won't have them. We wouldn't have the two secondary polar bodies. The first polar body, I mean, won't divide and give us secondary polar bodies. But we do have the secondary oocyte divides usually into fertilized egg and secondary polar body. So the number of secondary polar bodies is variable. It could be at least one or three. And only now, by the entrance of the sperm, the meiosis 2 has finished. And as you can see, this is something interesting. It's not written in any textbook, and this is an open question for all of you. Do female really have in their bodies haploid cells? This is a question for you to answer. Now this is a comparison between, I finished my lecture here, but I have to just remind you about the comparison between spermatogenesis and oogenesis. Spermatogenesis is straightforward. We have 4 equal in size spermatids, then they go and they form sperms. However, when we compare oogenesis to spermatogenesis, we have only one cell that has been really ovulated and carried on fertilization. How about these? Here, the secondary oocyte, it is big. Why? Because it's stolen the cytoplasm of the first polar body. And exactly the same would happen here. The fertilized egg would steal the cytoplasm of the secondary polar body. So if we compare the chromosomes of the secondary oocyte and the first polar body, they are the same. 1N, 23 double structured chromosomes. When we compare the fertilized egg with the second polar body, chromosomally they are 1N single structured. So the only difference between them is the amount of cytoplasm. The secondary oocyte steals the cytoplasm of the first polar body, and the fertilized egg steals the cytoplasm of the secondary polar body. So the outcome of spermatogenesis is huge number of sperms, and the outcome of oogenesis is only one egg. Why? Because the female, again, she would carry the baby, so she can't give more than one oocyte because she would have to carry it. Of course, twins and other twins and triplets, we can talk about them later on, different mechanism. And that's why the male has to take care about the number, because the male does not carry the baby, and this is something repeats itself in the whole animal kingdom. It would be interesting to also revise these two tables here. What else would happen to the oocyte once the sperm has entered it? It would be activated by the sperm, and the new life is given here. The zygote, the unicellular, the fertilized egg, the unicellular embryo would not stay rest. It would go into multiple division. We call them the cleavage, and that should be the topic of the next lecture. And here the main results of fertilization. We have zygote, diploid number. So we've gone through we went through meiosis to have haploid cells. Then we go through fertilization to restore the diploid number. Only now we can say genetically this is a male or female, and finally initiation of cleavage, which is the topic of next week. If there is no sperm, oocyte is not fertilized, the cell degenerates approximately 24 hours after ovulation. What does this mean? It means that if the female, she is not married, at least in our culture, and there is no sperms, all her oocyte, they will never ever reach there, and they will never ever finish their meiosis too. And this is very important. Females, they need the entrance of the sperm to finish their meiosis. On the other hand, males, they don't need the female to finish their meiosis too. Sperms, they ejaculated ready. Maybe they need the female reproductive system to remove the plasma membrane from the sperms to ovulate the egg. This is also something that does not make female or male having the upper hand on each other. However, female in the whole reproduction is carrying the baby, and it has the upper arm all the way through. So all the lectures about the oogenesis should be summarized here. In all textbooks for your future exams, this is what you need to know about oogenesis. But I'm sure that without the previous lectures you can't understand these simple marks here. And by this I reach the end of my lecture. See you next week.